Thank you, Ursula. So I'm going to start off, and I'm going to be talking about improving the predictions of the risk of emergence to resistance, which I think is very similar to what the other speakers have been talking about. And the take-home message that we're going to get to at the end is the same as the other speakers, which is context is important. It's not simple. There's a lot of things that go into trying to make the decision whether or not resistance is going to be a large problem for a program that you're working on. Okay, so I'm going to start off with a little bit of background, and I'll be focusing a little bit here also on the value of a multiple target versus single target drugs. So what is a single target antibiotic? A single target antibiotic is a drug that targets one gene product expressed by one gene in the TPP pathogens. So for example, the rifampicin targeting the RPOB gene product, the beta subunit of RNA polymerase, one gene, one product. So that's an example of a single target drug. And so what are the types of resistance that you can get to single target drugs? Well, you can get spontaneous resistance mutations that can be selected within a population, pre-existing mutations that are then selected by the presence of the drug. You can have pre-existing resistant populations that can be selectively enriched. So for example, if a particular TPP pathogen has multiple forms of the gene target, then if there's a resistant form, it can be enriched when subjected to selection by the drug. And then of course there's resistance acquired by horizontal gene transfer. Some complications. Some TPP pathogens may have more than one gene product, which is going to make things more complicated for selecting resistant mutants. And multiple targeting drugs may effectively be single target if the balance between the targets is unbalanced, and we'll get back to that later. And then, of course, resistance may be due to off-target effects, so efflux, entry, or horizontal gene transfer. So the background questions that I'm hoping to address in this talk are, do single-target drugs carry unacceptable risks of mutational resistance? And when we're talking about risks, the risks that you can be talking about are economic risks. So are these going to pose a risk to your drug that at some point you're going to decide to stop development because you decide that the risk of resistance development later on is too much of an economic risk to your project? For example, you don't want to get to the point of doing clinical trials only to decide that your compound has unacceptable resistance liabilities. And then further down the road, there's a medical risk, which is clinical failures. You don't want to be developing a drug that ultimately is not going to be useful because of its resistance liabilities. Now, can the risk of resistance be predicted, and which studies are important for this? So Michael's talk did an excellent job of going through several of these. So you have your studies to detect resistance, the emergence of resistance. So for example, frequency of resistance or serial passaging, and you can do single selection or multiple ascending selections, which would be the serial passage. He also addressed MIC90, which is you take a large pool of randomly selected clinical strains and you test the resistance to your compounds in that large pool, and the concentration that inhibits 90% of that pool is your MIC90. And that gives you an idea of what level of resistance exists out in the world already to your compound, and can be quite informative. And then there's analysis of the resistant mutants, which includes the level of resistance conferred by the mutations that they acquire and the fitness cost of those mutations. Again, things that were both addressed in the previous talk. Now, additionally, there's a question, does predicting mutational resistance matter? And it matters in the sense of choosing your TPP. What organisms are you going to target? Because I think that the resistance can really inform what possibilities you have for targeting different organisms. And then the economics of development. You know, is your chosen TPP, is it broad enough? Is there enough demand to make it economic to continue developing your drug? And then ultimately, underneath all of this, and I'm not going to go too much into this, is the unpredictability of horizontal gene transfer. That's both in the sense of whether there is resistance conferred by genes that are already out in the world and might be transferred into your pathogen, or even before that, whether there are genes that have not that are in a different environment, for example, the soil, that whenever you release your drug out into the world are now going to be selected to be horizontally transferred into your organism. And some of this you can look for example, in the McNineties, and some of it is a little bit unknowable until you've actually released it. Okay, so what's more important? Horizontally transferred resistance elements or resistance mutations? And both is the answer. It really depends on the context, going back to the context. So if you look at MTB, tuberculosis, the resistance is conferred by mutations in all cases. And if you look at Enterobacteriaceae, beta-lactamase, or beta-lactam resistance is all horizontally gene transferred, essentially. And then somewhere in the middle, you have examples of drugs and bug combinations where you find that it's a little bit of both. That, for example, fluoroquinolones and Enterobacteriaceae, resistance is conferred by mutations and horizontally transferred elements in some combination. Often both are involved. So it really depends on the exact context you're looking at. Okay, so this is to address the question of whether there are advantages to single drug targets. And in this list, I mean, obviously you can see that single drug targets confer an advantage in the one situation where you're looking at mutations that are actually on the target. Then if you target a single target, you expect there to be a higher rate of resistance mutations because you only have to get mutations in one gene versus a multi-targeting drug where you'd expect to see a lower rate. But in other resistance mechanisms, for example, resistance conferred by upregulation of efflux or alterations of influx, drug modification resistance, horizontally transferred generally, or target protection resistance, you don't really see any difference. So single target drugs have somewhat higher liabilities because of the fact that they only hit one target in the cell, but for many resistance mechanisms, that actually doesn't make a difference. So I don't think if you find yourself developing a single target drug that that's necessarily such a big problem. And we're gonna go on to talk about how multi-target drugs can oftentimes effectively be single target. Okay, so talking about the different types of resistance conferring mutations, both of the previous speakers mentioned various ways that you can select resistance mutations and a little bit of discussion of rates of mutation generation. And what you see is target mutations, so typically where you have a single nucleotide change that makes an alteration to the protein product. You find that the rate of any one of those mutations is 10 to the minus 10. Now that's 10 to the minus 10 number assumes that there's only one change that you can make in the protein that can confer resistance. And later on, we're going to look at several examples where it's really often there are many changes that you can make that can confer resistance. And in that case, the rate is multiplied up by the number of possible mutations that can get you to that point. Additionally, if you go down, the next item in the list is gene inactivating mutations. Here we have MAR and ACRR, efflux pump regulators, and E. coli, where you see the frequency is the same, it's 10 to the minus 10, but now you have a much larger target. That for one of these genes, the target is several hundred nucleotides that you could potentially mutate to inactivate any of these genes and reach a higher level of resistance. And then down at the bottom, we have duplications and amplifications, which are something that's not often considered. But when Michael was talking about the stability of resistance mutations, a lot of times the unstable mutations are amplifications or duplications. And you find this coming out a lot whenever you're doing selections against beta-lactam type genes, because amplification of beta-lactamases can oftentimes confer higher levels of resistance, sometimes enough to overcome the novel mechanism of a new beta-lactam type drug. And so those are actually kind of important, and I agree, it's quite important to test the stability of your mutations. But underlying all of this is the fact that it almost doesn't matter that the rate of mutations, there's good and there's bad, but ultimately you're never going to be so good that you're going to eliminate it as a possibility. You see at the top these these targets that the rate is 10 to the minus 10, that doesn't mean we don't see them. So even though some of these drugs have very good numbers, you're still going to have those mutations arising out in the world, and then it's a question of what those mutations mean. What is their fitness? What is their increase in resistance? And that's going to determine how important that frequency is. So moving on, I'm going to talk about several examples of drugs. Some of these have already been addressed, and I'm going to tell you now that I'm going to talk about them in broad strokes. So if I say something that upsets you because I'm not being technically correct, I'm sorry, but I'm trying to make broad points about these drugs. So we talked a little bit about rifampicin. So rifampicin is a single target drug. The mutation rate is about 10 to the minus 8, and that's because even though the target is a single target, there's more than 100 different mutations that can confer resistance. And the single-step resistance confers a high level of mix. So you go one step and you become extremely resistant to rifampicin. And with these levels, you expect that you're going to select for resistance in any particular infection that you treat with rifampicin. Most mutations have a significant fitness cost, but there's clinical and experimental evidence for selection of high fitness mutants. And there's an additional factor, which is genetic compensation of the highest fitness mutants. And so this is just one more complicating factor, if it wasn't complicated enough. That what you find is that when you look at rifampicin-resistant mutants out in the world, the ones that are already have high fitness, they're fine, and you find them as they are. But for the mutants that have a lower fitness, what you find is that they quickly acquire compensatory mutations to reduce the fitness cost of the mutations that they have while retaining the resistance. So the high fitness cost of the mutations doesn't really get you very far in terms of preventing these mutations from being selected and retained out in the world. And so the context for this is even with a high frequency of resistance, even with high fitness, even with high resistance, rifampicin has a clinically useful TPP, which is it's one of the most important drugs for treating tuberculosis. So this is only to say that the context of the drug you're using and where you're planning to use it in terms of TPP is really important. Another example that Mariana talked about at the beginning was fusidic acid. Again, a single target drug that targets EFG. And the mutation rate is about 10 to the minus 8, similar to rifampicin. Rifampicin has more than 100 potential mutations. Fusidic acid has around 25. And resistance is, again, expected during infection because of these high frequency of resistance that you see. Most mutations do have a fitness cost, and same as with rifampicin, you get compensatory mutations for those high fitness fusidic acid resistant mutants. And resistance is also conferred by horizontally transferred genes. And the high frequency of resistance of fusidic acid limits its use alone for now, and maybe that's wrong, but it can be used for topical infections where the concentrations that you can achieve make the resistance almost irrelevant. You can just overwhelm the resistance, even high level resistance. So at least that's one example with fusidic acid of where these high resistance, high frequency of resistance, high fitness actually can be overcome because of the specific context in which you're treating. Another example here is mecilinum, which is a beta-lactam type antibiotic, but is a single target, PBP2. This was, again, already talked about. The frequency of resistance is extremely high, and the resistance in clinical isolates is due to inactivation of cis-B, which results in upregulation of PPP-GP, and that renders PPP2 non-essential. But the resistant mutants do have a high fitness cost, and as a consequence, the majority of ESBL-producing Enterobacteriaceae are still susceptible to mecilinum. And mecilinum's not used here or broadly in Europe, but it's used in Scandinavia, and it has been used for quite a long time. And in spite of long-term use, there hasn't been development of resistance, indicating that indicating that this fitness cost may have a role in maintaining that the resistant mutants don't proliferate in the population. We don't see them expanding and taking over like we do with some other drugs. Aminoglycosides, most of you are probably familiar with aminoglycosides, and you see that the situation for aminoglycosides is quite different depending on the organisms that you're planning to treat. So if you look at gram-negatives, they tend to have many copies of their 16S ribosomal RNA, which is the target for aminoglycosides, which means any single mutation in one of those copies of the 16S doesn't confer resistance. You'd have to mutate all 7 of them at the same time. And as a consequence, you don't see mutations in the ribosomal RNA in gram-negatives. The exception is streptomycin, where you do get mutations that affect S12, but as a consequence, streptomycin is seldom used for gram-negative infections. But the observed resistance for aminoglycosides in gram-negatives is almost exclusively horizontally gene-transferred. However, with MTB, what you find is that there's only one 16S ribosomal RNA gene, meaning that you do pick up mutations in the ribosomal RNA genes, or the ribosomal RNA gene. And similar to the case with rifampicin and fusidic acid, the high fitness ones are then compensated for, meaning that all of the mutations that you see, whether they started off with a high fitness mutation, they eventually end up high fitness with high resistance. And there's no horizontal gene transfer in MTB. And then we have the case of fluoroquinolones. So fluoroquinolones are interesting because I've mostly been presenting drugs that are single target drugs. Fluoroquinolones are famous for being multiple target drugs, that they hit both DNA gyrase and topoisomerase 4. But the trick about that is that depending on the organism, it hits those 2 genes unequally. And so in some cases, for the first step mutations, you're hitting gyrase, and in some strains, you're hitting topoisomerase 4. And then what you end up doing is leapfrogging one above the other. So you get a gyrase mutation, now topoisomerase 4 is limiting, so then you pick up a topoisomerase 4 mutation, then you go back to gyrase being limiting, so you pick up a gyrase mutation. And so in this way, even though it's multiple targeting, essentially it can climb its way up to very high resistance. And key to the resistance of fluoroquinolones is also that the mutations that confer resistance to fluoroquinolones are very low fitness. So they, once they're selected, they tend to stay in the population because there's no selection for them to be swept out. Additionally, there's horizontally transferred elements that confer resistance to fluoroquinolones. And so in spite of this having very good properties, we do know that there's quite a lot of fluoroquinolone resistance out in the world now because of the fact that even though it's dual targeting, you can climb your way up to quite high resistance. Okay, so I'm going to sum up with a little bit of a summary slide here. So this is an image from a review. And on the left side here, we have relative fitness, and on the bottom, we have relative resistance. And these blue circles represent mutations arising, and the size of the circles indicate the frequency of the mutations. And so what's important to do when you're developing a drug is you really want to avoid the high-risk space, which is those mutations towards the top right, which are, have a very high fitness and a very high relative resistance. And this has additional things about the types of selections that give rise to those mutations, but it's really beware of the high-risk space. So you're a drug developer. What can you do? Most new antibiotics will be single targeting, and once you've started developing your compound, there's not a lot of options to change the target of it. So you have to deal with the compound series that you have. So what are the problems to look out for? Beware the combination of high frequency of resistance, high fold shift in MYC, and high fitness mutants. And I was thinking about it, and any combination of two of these can be problematic for you, but in particular, if the mutants that you find that you're selecting and discovering by whole genome sequencing, for example, if they're high fitness, that can be quite problematic because it means that they'll likely be selected and proliferate as opposed to being eliminated like some low fitness mutants likely will be. What can you do to mitigate that? I mean, it's a little bit trivial, but you can improve the potency of your compound, can overcome some of these problems if you can just get past the high level of resistance that it confers. Or, yeah, so I mean, make your compounds as potent as you can is sound advice. Measure the TPP MIC90. So look at the organisms that you're intending to target, look at broad panels, and see what's out there. If it turns out that your compound has a very narrow distribution of mix against your TPP, that's great. But then if you find that there are some resistant mutants out there, it would be good to try and figure out what they are. Michael addressed the fact that it can be quite difficult in resistant strains to do that, but it kind of depends. If it's something that's on the chromosome or if it's a mutation, you're stuck doing, as he mentioned, something like a genome-wide analysis study. But if it's a horizontally transferred gene, sometimes you can just look at the horizontally transferred elements that all of your high resistant strains carry and see what they share in common, and that can sometimes be informative. Another mitigation strategy that you can use is rather than going for a broad spectrum, you can consider a more focused or specialized TPP, which can sometimes allow you to overcome some of these problems depending on what you're looking at. So for example, we talked a lot about MTBs, and MTB's specific characteristics mean that some drugs that might not be suitable for treating a UTI can be quite useful in treating MTB. And then ultimately, what you want to get to is a go or no-go decision based on all of these experiments, as detailed by Michael. You gather as much information as you can, and then you just have to try and make an informed decision. There's not going to be a simple, you've checked a box and now everything's okay. It's more than likely that you're going to have to consider all of these things and make an educated guess about how big a deal resistance is going to be for your program going forward. Thank you.